Today, we are going to talk about membrane transporters in plants. Membrane transporters are proteins that mediate the entry and exit of substances through the plasma membrane. They are capable of recognizing and binding to specific substances, thereby facilitating substance transport. Membrane transporters are divided into different types based on the substances they transport. They are, for example, ion transporters, sugar transporters, amino acid transporters, and aquaporins. The membrane transporters also inhibit reactive oxygen species generation through ion regulation, sugar and amino acid transport, hormone induction, and other mechanisms. In addition, they promote enzymatic and non-enzymatic reactions in plants, also activate antioxidant enzyme activity, and promote reactive oxygen species scavenging. Membrane transporters in plants. Membrane transporters are embedded in the plasma membranes of cells and various organelle membranes and can be classified into different types, as I already told you, based on their transport characteristic for different substances. They are ion transporters, sugar transporters, amino acid transporters, and other compound transporters. First of all, ion transporters. Ion transporters include sodium ion transporters, potassium ion transporters, calcium ion transporters, hydrogen ion transporters, and anion transporters. First, we'll start with sodium ion transporters. Sodium is the most abundant type of cation in extracellular fluid, playing a role in maintaining cellular water and acid-base balance. There are two main types of sodium ion transporters in plants. First type is located on plasma membrane. Just look at this diagram. This is plasma membrane. And second type is located on vacuolar membrane. This is vacuole, right? The first type of sodium transporters, which are located on plasma membrane, they control the transport of sodium across the plasma membrane. Influx or efflux. Influx of sodium ion is controlled by high-affinity transporters, HKTs, number one, this one. Low-affinity transporters, LCTs, number two. Nucleotide-gated channel, this one, CNGCs, and ionotropic glucose receptor, GLR channels. The efflux of sodium ion is controlled by salt overlay sensitive 1, SOS1. SOS1 is a sodium hydrogen ion antiporter pathway. They control the transport of sodium from the cytosol to vacuole through the exchange of sodium and protons. Then second type, which are located on the vacuolar membrane and controls the transport of sodium across the vacuolar membrane, right? This is vacuolar membrane. In addition, some studies have indicated an interaction or relationship between sodium and proton transporters and salt overlay sensitive 1, SOS1. Second type is potassium ion transporters. There are many potassium ion transporters in plants, including HKT, means high affinity potassium ion transporters, KT, HAK, KUP, then AKT, then 2-pore channels, TPCs, and cation proton antiporters. These transporters are distributed on the plasma membrane and vacuolar membrane and can transport potassium ion under different conditions. HKT was identified as high affinity potassium ion transporter protein, that is an alkaline cation transporter linking cytosolic osmotic homeostasis with plant tolerance under salt stress. The KT-HAK-KUP transporter family belongs to the amino acid polyamine organocation superfamily. It regulates the transport of potassium ion in low potassium ion concentration environments and is involved in the redistribution of potassium ion to maintain sodium-potassium ion levels. The AKT family includes AKTs and HKTs, which are potassium ion channel protein. In addition, two types of cation proton antiporters, HK and KEA, can also provide additional potassium ion transport capacity in high concentration of potassium ion environment. But their transport mechanism is unclear. Next is calcium ion transporters. Calcium is an essential nutrient for plants. Calcium ion homeostasis is highly important for maintaining the integrity of cell membrane structure and for maintaining intracellular enzyme activity, like sodium ion. Calcium ion membrane transporters are located on the plasma membrane and control the transport of calcium ion across the plasma membrane. The influx of calcium ion is controlled by mechanosensitive channels, OSCAs, then CNGCs, GRLs, GLRs, and TPCs, right? OSCA1 can play a role in osmotic stress, and TPC channels can specifically mediate the influx of calcium ion. The efflux of calcium ion is energy dependent and is mainly achieved through calcium ion ATPase, right? Another type of calcium ion membrane transporter is located on the vacuolar membrane and controls the efflux of calcium ion from the cytosol to the vacuole. Calcium sodium ion exchange regarding mechanism. Calcium sodium ion exchange can transport calcium to the vacuole through the exchange of calcium and sodium, and calcium proton exchange can transport calcium to the vacuole through the exchange of calcium and proton. Next is hydrogen ion transporters. Proton ATPase and proton PPase are involved mainly in transport of hydrogen ion in cells. Hydrogen means proton ATPases are divided into plasma membrane proton ATPases, PMAs, and vacuolar membrane proton ATPases, VMAs, right? Actually, hydrogen atoms lose electrons to form proton, which can regulate the pH inside plant, promote plant growth and development, and improve nutritional quality. PMAs can generate a proton gradient, which drives SOS1 to transport sodium ions. VMAs and V proton PPases are located on the vacuolar membrane and are responsible for transporting proton from the cytosol to the vacuole. These two types of transporters generate proton gradients on the vacuolar membrane, then driving VHX to transport sodium ion, right? Next is anion transporters. Inorganic anions in plants include chloride ion and nitrate ions, which are regulated both inside and outside the cell by two anion channel proteins. Slow anion channels, SLAC or SLAH, and chloride channels, CLC. SLACs can regulate the distribution of anions in the xylem in the extracellular space. CLCs regulate the transport of anions through their intracellular compartmentalization effect. In addition, aluminium activated malate transporters, ALMTs, are distributed on the plasma and vacuolar membranes and are involved in the transport of chloride ion. Other ion transporters. The iron nicotine amine transporter, yellow stripe like 2, OSYSL2, is responsible for transport of iron, right? Metal tolerance proteins, MTPs, control the transport of zinc ion and are associated with zinc sensitivity and tolerance. Vacuolar ion transporters, VITs, control the transport of iron, zinc, and magnesium ions. Cation diffusion facility, CDF transporters, transport heavy metal ion like cadmium ions, cobalt, and nickel through the exchange of cations and protons. Iron regulated transporters, IRTs, control the transport of cadmium and nuclein in plants. Natural resistance associated macrophage proteins, NRAMPs, are located on the vacuolar membrane and transport cadmium to the vacuole for chelation. ATP binding cassette, ABC, transporters are the most ubiquitous in plants and are currently the largest family of membrane transporters. Next is sugar transporters. After ion transporters, second type is sugar transporters. Sugar are important component of plant cells and occur in the form of sucrose, fructose, glucose, starch, and other substances in the plant cells. And we know they are responsible for energy supply and signal transduction in plants, right? There are three main types of sugar transporters in plants. Sugar transporters, SUTs, then SWEETs, sugar will be exported transporters, and monosaccharide transporters, SMTs. SUTs are located on the plasma membrane and are only found in plants. SWEET are distributed on both plasma membrane and the vacuolar membrane. MSTs belong to the major facility superfamily, which consists of 7 subgroups: early response to dehydration, sugar transported proteins, plastic glucose transporter, inertial transporters, vacuum glucose transporters, tonoplast sugar transporters, and polymer monosaccharide transporters. They are distributed in different locations, controlling the transport of sucrose, maltose, glucose, sugar alcohols, and other sugars. Next is amino acid transporters. Amino acids are key nutrients required by plants and play an important role in promoting plant photosynthesis and material metabolism and enhancing plant tolerance. The amino acid transporters, AAT family, can be divided into two categories: the amino acid polyamine choline transporter, APC family, and the amino acid oxine permease, AAAP family. The APC transporter superfamily include cation amino acid transporter, CATs, polyamine proton cotransporters, PHS, and amino acid choline transporters, ACTs. KATs control the bidirectional transport of GABA, glutamate, and aspartate between the cytosol and vacuoles. PHS mainly play a role in polyamine transport. ACTs control the bidirectional transport of GABA between the cytosol and mitochondria. AAAPs include amino acid permanence transporters, AAPs, lysine histidine transporters, LHTs, proline transporters, aromatic and neutral amino acid transporters, putative oxime transporters, GABA transporters, etc. The AAAP family plays an important role in the transport of GABA, lysine, histidine, proline, and many other amino acids. Thanks for watching. If you liked this video, make sure to subscribe for more. Thank you.